3D imaging, memory manipulation, energy storing solar cells, and a Nobel Prize for super-resolution microscopy.
That's this week on Light Matters. Hi, I'm James Lowe.
And I'm Laura Marshall, and this is Light Matters for October 15th, 2014. Software that uses video gaming gear is bringing live cells into a 3D spotlight. Researchers from Drexel University are developing a 3D software program called Lineage Editing and Validation, or LEVR 3D, to identify, tag, and track live cells. The new system will capture patterns of motion and cell division using sequences of microscopic time-lapse images. Such enhanced imaging produces a 3D rendering of the cells. A stereovision projector was able to bring the 3D image data to life. LEVR 3D compiled data from the multi-layered microscopic images while the researchers used 3D gaming glasses to look at the microscopic cross-section. The LEVR software can delineate cells, color code them, and denote the exact moment of their division. It has proven more accurate than conventional methods and can provide more data than manual processing, according to the researchers. They hope to enhance existing visual data to facilitate identification of cell changes over time, which is key to studying the abnormal cell proliferation that causes cancer and also when using stem cells in regenerative medicine. The research was published in BMC Bioinformatics.
Optogenetics is giving scientists a better look at how different parts of the brain can work together to manipulate memories. Using the light-based technique, researchers at the University of California, Davis have been able to erase specific memories in mice. Their findings prove the basic theory of how different parts of the brain will work together to retrieve episodic memories, they said. The researchers used mice that were genetically modified to make their nerve cells fluoresce green when activated by light, and to make the cells express a protein that allows them to be switched off by light. The mice were trained by placing them in a cage where they get a mild electric shock. Normally, mice placed in a new environment will nose around and explore, but when placed in a cage where they have previously received a shock, they freeze in place in a fear response. The testing first showed that the researchers could label the nerve cells in the cortex and hippocampus that were activated in learning and memory retrieval, and then that they could also switch those memories off with light directed through a fiber optic cable. When they did this, the mice behaved in a normal way again, showing that they had lost their memories of the unpleasant event. The investigators also demonstrated that turning off other cells in the hippocampus did not affect the retrieval of those memories. Optical fibers can subsequently be traced from the hippocampus to specific cells in the cortex.
Solar cells could soon double as rechargeable batteries thanks to work by a team at The Ohio State University. The researchers' new hybrid device combines a battery with a solar cell, potentially reducing energy costs by at least 25%. With this device, light is converted to electrons inside the battery, allowing nearly 100% of the electrons to be saved. The researchers designed a permeable mesh solar panel from titanium gauze using red dye to tune the wavelength of light it captures. Air passes freely through the gauze while the rods capture sunlight. The mesh solar panel then forms an electrode. Beneath that, the researchers placed a thin sheet of porous carbon and a lithium plate. This hybrid design uses 3 electrodes rather than the 4 usually needed to connect a solar cell to a battery. Between the electrodes, the researchers sandwiched layers of electrolyte to carry electrons back and forth between the battery electrode and the mesh solar panel. Based on early tests, the researchers said the new solar battery's lifetime could be comparable to rechargeable batteries that are already on the market. The research was published in Nature Communications. And now over to James with news on the Nobel Prize in Chemistry.
Thanks, Laura. On last week's show, we told you about the Nobel Prize in Physics, which was awarded to the inventors of blue LEDs. Now there's more big news to report, as the chemistry Nobel winners were announced the next day. Eric Betzig, Stefan W. Hell, and W. E. Morner were awarded the Nobel Prize in Chemistry for their work on circumventing diffraction limits to achieve nanoscale microscope images. Hell, director of the Max Planck Institute for Biophysical Chemistry in Germany, was recognized for his development of stimulated emission depletion, or STED microscopy, which uses one laser beam to stimulate fluorescence in molecules and another to cancel out all fluorescence except for that at the nanoscale. Working separately, Betzig of the Howard Hughes Medical Institute's Genelia Farm Research Campus in Virginia and Morner, a professor at Stanford University, were recognized for development of a single molecule microscopy, a method that relies on turning individual molecules' fluorescence on and off and imaging with nanoscale detail. All in all, I'd say the field of photonics has had a pretty good week.
Yes, it certainly has, James. That's it for this week's show. Email us your questions or comments at lightmatters@photonics.com. And don't forget to follow us on Facebook and Twitter for news updates every day. Thanks for joining us this week. We'll see you next time.